So, if you could just tell us a little bit about your research, what you're doing and why.
Yeah, I do research in lipid membranes that incorporate ion channels.
Okay.
And the reason for that is that they are tools to make very miniaturized nanostructured devices that can be either biosensors or, in our case, energy producing devices. So the basic research is to look at how ion channels and transport proteins incorporate into lipid bilayers, which we can control a lipid bilayer to be a tethered or a supported lipid bilayer for stability of the system. And then with the ion channel or the transport protein, we incorporate those proteins into the tethered or supported lipid bilayer to create a nanostructured device that's stable enough, stable meaning for weeks or months or years, hopefully, that can then produce energy in the same way that cells of the body do. So that's the dream, and it's some of the early results show it's kind of reality as well.
Okay. And that's what you're doing. So why would you do something like this?
To create alternate energy sources, and with the main aim for implantation.
Okay.
So at the moment we have experiments where we're using glucose biofuel cells in animals to replace lithium batteries, and now and what we're doing with the lipid bilayer membrane channel work is to create a biomimetic approach to a fuel cell, and that is the target then is to be able to generate enough power that we can replace a lithium battery for our first medical device target as a pacemaker. Yeah, so that's and so it's a kind of a dream, but the dream has turned into reality. I've been in France now for 4 years, and the first 3 were as a char d'excellence that allowed me to produce a prototype that worked. And now the French government has, with 2 large grants, have provided the funding to build a team in Grenoble to carry this forward and we're carrying it forward with a industrial partner, Sorin, who's one of the company makes pacemakers worldwide.
Okay, great. So what kind of techniques are you doing in the lab that will enable you to carry out this research?
Yeah, I mean the fundamental research is with ion channels and lipid membranes. So core to this is patch clamp electrophysiology.
Right.
Yeah,
okay.
so that's really core. The other things that are useful to us are electrochemical techniques. We have a nice potentiostat, and we do electrochemistry on the bilayers, looking for conductance. And so we're manipulating we're manipulating with the potentiostat to be able to measure conductance in a very simpler way. So that's for screening. But so the tools are really electrophysiologically based, patch clamp, electrochemical, and the standard tools that you'd see in an electrochemical laboratory.
And which samples do you typically work with for the patch clamp?
Yeah, with mainly liposomes and channels expressed into liposomes. So unfortunately we haven't patch clamped many real cells recently, but so most of the work is done with purified ion channels that then we incorporate into liposomes that we can patch clamp and measure the properties of those transporters, and they're the transport proteins that we need to know precisely the characteristics so that we can use those in the biomimetic devices that go into the tethered and supported lipid bilayers.
Okay. So your future ambitions, you mentioned it briefly earlier, where would you like to be? How would you like this to be going in the future? What's your goal?
Well, I've created a rod for my back because the major grant that started this year is due for completion in 4 years' time.
Okay.
So the medium-term goal is in 4 years to have an alternative power supply that can be implanted into animals at the moment, but looking then for implantation into humans. So we're going along that track quite smoothly. So the medium-term ambition is really implantation into animals.
Right.
Longer-term ambition, implantation into humans, perhaps between 5 and 10 years into the future. You know, we could get lucky. Yeah.
So how did you hear about Scientifica?
Yeah, it's an interesting story which I'll share with you. I've been working in Australia for quite some time, and then moved to France in 2008, middle of 2008, and I was looking for a place to buy patch clamp equipment to re-establish a laboratory in France, as I had in Australia. And I came across Scientifica, and it was a kind of a one-stop shop for me. So I had a very nice Olympus microscope I could buy, and the Faraday cage I was used to, and the patch clamp amplifier I was accustomed to, Axon. And the one thing that I was really surprised and happy about was the manipulator, the patch style manipulator, because I hadn't that wasn't available to me in Australia. I found it was available to me in France, and it's very stable, much better than my Narashigi oil-filled systems. Yeah, exactly. So yeah, I'm very happy with the being able to establish the patch clamp system based on Scientifica.
And what year was that, did you say?
The end of 2008, really.
Okay.
End of 2008, beginning of 2009.
And you're still using them now, and they're still performing as they should?
They haven't broken. No, it's all performing quite well, I must say. Yeah.